as you have alluded, the title of the overall workshop is Metabolism and Mathematical Models, but I put it modeling, 2 for a tango, but maybe 3, I would argue. For example, it's metabolism and qualitative modeling. So in the aspect of qualitative modeling, we have the question, given a set of enzymes, for example, how many reactions can evolve or can we design? There is also metabolism and structural modeling. Given a genome, how many reactions are there and how are they organized in functional networks? And of course, the metabolism and quantitative modeling, as Ina discussed earlier, given a set of omics data, how metabolic fluxes distributed, how metabolism responds. So I will start giving you this kind of perspectives from our work, which of course is based on the work of the whole community on these 3 particular aspects. So in the area of qualitative modeling, so what do we perceive as qualitative modeling? Of course, we all know the biochemistry. These are the maps that you see in databases like ECole. By now, it's very well organized in reaction in metabolic maps, in pathways, and also all the way down information about the enzymes. And of course, as we all know, the area of metabolism, there is a growing domain of applications already for years, agriculture, metabolic engineering, all the way to drug design and toxicology. But what are today's challenges? Today's challenges, I think it can be summarized really in this graph, where you can see that over the years, how the information in databases have evolved. So we continuously increase of reactions and continuously increase in compounds that we don't know what are the reactions they participate. And for the reactions, sometimes we don't even know what are the gene sequences they encode for. So the question now here, that as we venture in these uncharted territories, how can we distill the spirit of biochemistry and try to navigate this complexity? And of course, coming from the area of computation, we ask the question, can we move from in vivo to in silico, and what are it takes for this? Towards this, we've been developing now for more than 20 years a method that we call BNICE.ch, which is Biochemical Network Integrated Computational Explorer. Actually, this is the work that I counted sometime that is work worth more than 100 full-time equivalent years of researchers at every different level, from master's students to PhD students to postdocs and some professors. So, but where does everything started? Everything started on the key observation that if you look at the enzymes, and probably you're familiar with this, there is the so-called enzyme commission classification system, where there are 6 major classes. Now there are 7, including the transporters. And then there is a second class, subclass, which refers to the functional group. And then there is the third class, which refers to the cofactor. And turns out that the 4th class is very specific to the substrate. So in this respect, now, by this observation, then we went zoomed in these reactions, and we also observed the following. So if we look at the reactions 2.7.1, classified as transferring phosphorus containing groups. That's a classification of the group the class 2.7.1. And these are 4 reactions that participate there. So one can observe that effectively all these reactions, although they share a different structural substrate, if you look at the substrate as a global molecule, in reality, the enzyme acts on a very specific reactive site, as we call. So based on this, we came up with the idea to define what we call the generalized enzymatic reaction rules. So enzymatic reaction rules essentially encodes this chemistry, and then based on this, then we take the information from the different that we have from in vitro and in vivo, and then we encode this in the computer. I will spare you the details about the way we do this encoding, but right now we have nearly 500 rules times 2, because they are bidirectional, one for one direction, the other for the other direction. And so now what the method does is we can sit in a molecule, and then we apply all the rules there. And then suddenly we can see that reactions are emerging where these rules, they identify the reactive site, and then they break and make bonds according to the corresponding rule. And this is what we call the first generation of the reactions. So these are essentially reactions, these are molecules that are predicted to be one step away, given an EC number of the third class, 4.1.1, 2.1.1, etc. Now, then we continue, we take these output molecules and we have the second generation and so on and so forth. So in this way, essentially, we can evolve starting from a set of molecules and a set of cofactors and a set of generalized enzyme rules to evolve everything possible that can be produced with some starting molecules upon successive generations. And then, of course, in these molecules, we have biological molecules, but we also have molecules that are chemical molecules we can find in the chemical database, have not been found biological database, and also some novel molecules. Now, can we create a global predictive network of biochemistry? Towards this, we decided to start building what we call the Atlas of Biochemistry. So in Atlas, essentially, we start with the KEGG database, and then we take all the compounds that are in KEGG, and then we apply these rules, and we say how many ways I can increase the number of reactions that connect these compounds without increasing the number of reactions. So we moved essentially from the KEGG. This is how the KEGG representation looked like with the reactions and the different compounds. And you can see here a lot of what we call orphan compounds. These are compounds that are not connected by any reaction, but they're just in database. And then when we applied these rules, not only we increased the number of the reactions there, what we call novel reactions, by 100,000, more than 100,000, but we also been able to connect these orphan compounds. And then, these are some general statistics. And then, of course, sky is the limit. Say, what is the next step? So the next step is Atlas X in order to start now looking for other databases. So we went and we looked at other biological databases. We selected and curated these databases, and we came up to have 1.5 million compounds. And with there, we had 56,000 reactions. And now what we can do, then as I presented you before, now we have the KEGG Atlas here, where it uses only the KEGG compounds. And now our Atlas X now includes other biological compounds that has bioactive molecules integrated. And ultimately, what we'd like to do is to move and now ask the question, given the chemical databases like the PubChem, where we have millions of compounds, how many possible enzymatic reactions we can foresee that we can connect? And how can we use this? Then, of course, we can use this for metabolic engineering, synthetic biology, also for synthesizing from biological man-made chemicals. Now, I will move on then to the case of the metabolism and structural modeling. So you are probably familiar for this, which is the action of what we call reconstruction of GEMS. I apologize for just the acronym. This stands for genome-scale metabolic models. So how do? We start in general with some genome sequencing. We made the reconstruction. Then by genome annotation, we identify what are the reactions present in an organism. And then based on this, we reconstruct the networks and we build the mass balance equations based on which give us the stoichiometry of the compounds. And in our case, we're using a lot of methods developed by Jens Nielsen's group and by Kiran Patil's group, Raven and Carvme, and also any kind of updates. And then, of course, we take these draft models because, and we ask the question, can these models reproduce certain physiology? What is the physiology? Can we capture the growth assays based on different substrates? And also some of the metabolites, metabolomics or fluxomics information. And actually, this turns out to be the rate limiting step, which is essentially how can we now reconcile the first pass of the drafts that we have for the genome scale metabolic models, the networks, in order to be consistent with the growth assays. So towards this, we have developed a method that essentially what it tries to do is to characterize the gaps in the GEMS. What is a gap? A gap essentially tells you that if in a growth assay I have a growth, but in my model predict no growth on this particular substance nutrient, this means that I'm missing some biochemical reaction. So what we did, develop this method we call nice game, and because it's based essentially uses the databases that we have also enlarged, the KEGG and all biological database, and in addition even novel reactions that we have from the Atlas X, and then we try to reconcile the false negatives, find the reactions inside these databases that can essentially reconcile. But also, it's not simple this. So we have multiple ways to look at the problem. So what is the situation? So we can have multiple substrates that they give certain behavior versus the data, but we can also have alternative reactions that can recover the observed physiology, that can fill the gap. So based on this, then we can just start choosing the reactions and the networks that are mainly that can be used for filling this particular gap. So in this way, we say we have the updated kind of curated model. Now, of course, there are many uses for these gems, like knockout essentiality predictions, analysis, prediction of flux profiles, evaluation of max theoreticals, yes, but also study of fundamental yet important application questions, like what shaped the evolution of NADPH reactions. I think I went into automatic mode. So what shaped the evolution of NADPH reaction specificities in École? And this is something we're all looking forward to hear from Stefan. So, but I'm going to also show you how we are using these approaches within NCCR microbiomes, which is effectively a national center in Switzerland, which is focused on the microbiome analysis. So this is over Switzerland. It's a big, more than 22 partners across Switzerland, hundreds of students and postdocs. And the idea there is to study essentially microbiomes in all aspects and from all different ways. So another question is, what are the microbiomes in the gems? How are we doing this? So I'm going to show you here for some of the gems we have there and how we are using them. So here I show the people from our lab and also other collaborators with whom we are working and building different gems. And these gems also, right now we have more than 200 gems for more than 224 organisms in the phylosphere of Thaliana, Rapidopsis thaliana, 23 organisms for synthetic soil community, for the honeybee gut microbiome, pathogens, and then also we're looking now to increase in all these different directions. So in one case that we study, for example, in the Salmonella typhimurium pathogen metabolism, so we try to identify based on growth assays. We try to make sure that our models capture all the biochemistry of the compounds that exist in the gut, so to be able, so that when we will try to make inference about these models in the presence of the marine gut, we still have the biochemistry as it's observed, at least by this in vitro experiment. So this is one part that one can see using. And of course, then once you understand the metabolism, maybe you can find targets. Maybe you can also understand the competition or out competition of the native microbiome. In another work recently published, worked with the group in Julia Vorholt in ETH Zurich, where we worked on developing or curating and developing the genome-scale models for 224 organisms that live in the phylosphere of Rapidopsis thaliana. And then based on this, now we can able to predict positive and negative interactions between these organisms. And most of them, almost all of them, have been verified also experimentally. So this is how one can use, this is what we call the qualitative, the structural kind of modeling. I'm sure you're familiar with this. I gave you a little bit of a flavor here. So, and second is metabolism and quantitative modeling. So given a set of omics data, how metabolic fluxes are distributed. So for this, and based on the comment I saw in the workshop of interest in parasitology, I decided to present to you some of our work that we're doing here in a consortium with people from the University of Bern, mainly Professor Volker Hosler and Professor Philipp Olias, now in Germany. They set them to study the using these chemoinformatics approaches, how one can target the apicomplexan parasites. So you're probably familiar with it. So what happens, you have the Plasmodium falciparum, which attacks the organism. Then if we would like then to understand what are the host pathogen metabolic interactions, next to understand what is the contribution of the host cell metabolome to the parasite nutrition, and how the waste products of the parasite are used by the native metabolism. And then we want to ask the question, which are the metabolites that are essential for the parasite but are dispensable to the host? And then how can we develop now targeted therapeutic strategies that we can target metabolism and so compromise the ability of the organism to grow? So for this, I'm going to present you what are the systems biology approaches we use to study these host-parasite metabolic interactions. We start again with metabolic models that represent the host, the parasite, and their interactions. We use experimental data from physiology, and then some computational methods to simulate the fluxes, integrate data, and then to analyze the physiology. Again, we start with the genomes, and we build now the models for the hepatocyte and the parasite, and then we put them together and try to understand how do we do this. So first, we start, I'll show you how we kind of build the hepatocyte specific model for metabolism, the parasite metabolism, and then the host pathogen interaction. So how are we going to go modeling the metabolism of hepatocytes? So we start, of course, based on the latest, or one would say one from the last best curated human metabolic model, Recon3D, which has nearly 6,000 metabolites and more than 10,000 reactions. All those are mapped to 2,200 genes. And of course, it has compartmentalization, etc. So, and then the next thing is that how can we use now experimental data in order to take this generalized genome scale metabolic model of metabolism in the human and make it tissue specific, or excuse me, or as we say, context specific. So where do we start? We start with some physiology. So based on experimental data, we have, let's say, that we estimate the growth rate of the cell. Then we look at previous work in modeling and also specific analysis, experimental, how the medium composition should look like. And these are the kind of basic media composition. And then we put all this together. And then we also, of course, impose that we have to have 37 exchange reactions which correspond to this medium composition, and then some other metabolites that are detected in metabolomics, exometabolomics. So we go to human protein atlas, we take transcriptomics data for these genes, and these are transcriptomics data from normal liver. So once we take these transcriptomic data, we see that they correspond to 2,053 metabolic genes, which means that essentially we have to have this metabolic genes present. The others must be absent or very, very low activity in order to have a network which we're going to call hepatocyte model. And of course, we keep the reactions that all the reactions that, as I said, that are expressed also for transport reactions to allow additional transport from the media for more complex media. And then also we identify what is the minimum of them are required for growth, given a certain medium that we have. And then we end up now with the hepatocyte model that has 2,200 metabolites, which is comparable, which are much less than the previous total model. It has 4,000 reactions and 1,500 genes. So now we have a model, a genome scale model that is specific to the hepatocyte. Next is how do we go for the Plasmodium falciparum, the malaria parasite that infects the liver. So for this, we have in the past developed a highly curated, also with thermodynamics information about the falciparum metabolism. That is around 1,300 metabolites, 1,300 reactions, and 300 plus genes. Of course, we model also compartmentalization and extracellular environment. And then we define now the nutrients to which the parasite has access in the liver. Because now we say, okay, if the parasite model has to be in the liver, then it has access to all potentially the metabolites that are in the liver. However, based on experimental and experts' knowledge, a lot of discussion with our collaborators that were experts in this field, we said, okay, these are the possible 241 extracellular compounds from the whole thousands of metabolites in the human cell that can be potentially be uptaken or transported by different mechanisms into the cell. And then again, 165 of these metabolites turns out to be in the liver model. So in other words, now I have a parasite that can live in a liver environment, and it can have the choice now to grow on 165 metabolites. Now, the question is, how are we going to go to define what are the different metabolic states of the parasite, and on the way, probably reduce the complexity of the parasite metabolism in order to put these two metabolisms together. So this is the first case that we ask the question, okay, so if I look at what my parasite can take from out of the 165, let's say, what is the minimum type of nutrients, substrates that it can uptake and what is the minimum set of products that it can secrete in order to grow to a certain growth. So this concept is, if you are familiar, is very similar like the extreme pathways or the elemental flux mode. So if we find, essentially, it's like we find an elemental flux mode or an extreme pathway for the parasitus, for the parasite. And then we take all this network, this global network that is in the system, and then what we do here, we collapse it into a single reaction equation. So this reaction equation now, then we add it into the liver model, and this way we can simulate the growth and the growth of the parasite and the equivalent draining of nutrients and production of different secretions. And this is an example of a parasitism. It's a bigger question. We estimate also you calculate also what are the stoichiometric coefficients. And it turns out that we need 32 substrates, and also it produces 26 products that are secreted in the cell. Now you can immediately see what is the interactions. Right now, the simple parasite with the minimum type of metabolic interactions still sucks 32 substrates and gives out 26 additional metabolites into the system. And of course, one can now formulate the problem and ask, do I have any other alternative set of 32 substrates and 26 products in total? And turns out that yes, we can have up to 112 alternative parasitisms. Again, it's like we have 112 elementary flux modes or extreme pathways, depends how you want to see it, which are the different ways that now in a single or combination we can see these parasites can grow in the cell. And of course, it's very interesting because there are certain substrates that are common, that's always required for the growth of the parasitism, which is again very useful information because we know what are the essential nutrients for the parasite. And then there are some others which are alternatives. And this is a kind of a distribution of how the different nutrients are, the percentage distribution across the 119, 112 alternative parasitisms. In the same way, as you would expect, there are certain secretions that are always have to happen in order to have a functional growing parasite, no matter what is the set of nutrients it grows. And then we have alternative secretions, which also makes sense that the alternative secretions are more than the alternative nutrients. And this is a kind of snapshot of how the different nutrients are used in percentage by the different metabolic states of the parasite. Now, once we have this, now, what can we understand the host-parasite interactions? Now, we take these reactions, we take the parasitism reactions, we add them into the model, and then we ask the question, what is the effect if I force now my parasite to grow into the cell to a certain amount? What are the uptakes of the nutrients that it get, and what are the products it secrete? How, let's say, growing of the parasite within the cell compromises the growth of the hepatocyte, and all these interesting questions. And of course, then you can ask knockout questions, etc., etc. Now, the question we would like to ask, though, and our collaborators would like to know, is how do the host metabolic pathways change in order to sustain the pathogen survival? Because what is observed is once we have the infection, especially in the very, very first, weeks, initially you don't even have any phenotype of disease because your cells, your cells or the host cells don't somehow they adapt in order to sustain this parasite survival without themselves suffering. So what we do is, our collaborators they use HeLa cells, infection and non-infected control. This is the typical system they use. Then they do transcriptomic analysis, and what they find which are the enzymes that go up and which are go down. So now the question will have, okay, if we look at any transcriptomic data, essentially the hypothesis usually is that if an expression of a certain enzyme, RNA expression of a certain cellular process goes up, this means that the activity of this process, whatever is this process, is also upregulated. That's a common kind of interpretation that you do. But in most of the cases, what the people do, they do point-wise analysis, or in the best case, they do gene enrichment analysis. But now in our case, we say, okay, if we ask also in the case of these changes to have these local changes to be consistent with the flux balances, can I have a consistent way to integrate all this information? Let me explain to you here. So essentially what we get the two models. We have two reaction networks. two gems that are characteristic, is exactly the same structurally. And then I have expression ratio about the different transcriptomic conditions between the condition 1 and the condition 2, or condition 2 over condition 1. And then I say, okay, if I force now my solution of the fluxes to be in the same ratio, not exactly the same, not 1-to-1, but if the ratio in the change of the fluxes in the two models solved simultaneously, how many of these assumptions can be consistent? And what is the consistent? That essentially that if I enforce a twofold change, for example, in one expression of a certain enzyme, then can I achieve near twofold change in the corresponding flux? And now think that you do this essentially for all the hundreds of enzymes for which you have expression data. And then in this case, we find, for example, in this case, so for infected cells, hepatocyte cells, we find that we have 700 deregulated genes, 94 up and 600 down. And when we ask the fluxes between the two models to follow this up or down kind of pattern, then we find that we have 698, so of course a smaller number, that can be consistently achieve this. But the nice thing with this is that we can also ask the question, is there any combination, alternative combination of consistency? So essentially now I still have 9 sets of 698 ratios of fluxes that are consistent with the enzyme expression data. And then I have some of them that are always kind of consistent and some of them that are changing between the cells. So this way we can understand in a very efficient way now on the flux level by doing this comparative kind of flux analysis that we can get an idea which are the pathways that are better deregulated. Because now what happens, this is the big deal here. Although we have information only about 707 enzymes, the fact that we use the genome scale, the balances, the mass balances for thousands of reactions, then I can even get some inference about the changes in metabolic fluxes near or far away from where the point of change or the points of change are. And now, of course, now after we have this, then we can ask the question, how does the parasite change the nutritional profile of the host? In this case, for example, these are some of the compounds that we found that the infected cells increases in order probably, and this is now under investigation when we will look closer analysis in the parasitosomes. So we will probably are increased in order to meet the needs of the parasite or to substitute for what the organism is losing towards the parasite. Now, of course, quantitative modeling, there is other aspects. Another big question, given a profile or distribution of fluxes, which are the rate limiting steps? How fluxes will respond to changes in enzyme activities in the environment? How flux metabolites will respond to changes over time? What are the dynamics, essentially, that also Ina touched upon? So, in this case, of course, we're talking about metabolism and kinetic modeling, but given the fact that I'm limited in time, I will leave this for some other time for you. And for now, I would just like to thank you.